Welcome viewers. In this lecture, I am presenting one of the popular topic of zoology, that is induced breeding in fish. This presentation has been prepared as a part of free online learning resources. Through this presentation, an effort has been made to explain various aspects of induced breeding, such as basic concepts of induced breeding, historical aspects of induced breeding, importance of induced breeding in modern aquaculture, factors influencing spawning in fish, and induced breeding techniques such as band breeding, striping, hypophysation, use of natural and synthetic inducing agents. I hope that after completing this presentation, you will be able to have a preliminary concept on induced breeding techniques and their various advantages and disadvantages. Let us have an introduction. As you all know that Indian major crabs and Chinese crabs necessarily breed in rivers during monsoon because during monsoon several physico-chemical parameters which are essential for breeding in fish are active in riverine condition. In ponds, although the carpfish get sexually matured, but they never breed in confined water like ponds because in pond environment, physicochemical factors essential for breeding in fish are not available. What is induced breeding? Induced breeding is a technique by which the economically important fish are bred through artificial stimulation. Broodfish are stimulated by pituitary hormone or synthetic hormone to breed in captive condition. Broodfish means sexually matured fish. In induced breeding, some of the methods employ artificially created natural environment for breeding in fish. One of the such type of breeding is band breeding. Let us have a look on history of induced breeding. The modern technique of induced breeding was first evolved in Argentina after the discovery of pituitary gland extract by Hauze in 1931. They did the experiments of induced breeding in viviparous fish using the pituitary extract. In 1934, Brazilians were the first for successfully breeding their indigenous carpfish by the hypophysation technique. The same technique was followed by United States and Russia. In India, the first attempt of induced breeding was made by Hamid Khan in 1937 on Indian major carp, Chihinus mrigala. In between, in 1955, Sudhuri tried the breeding of Indian minor carps, namely Isomas dendricus and Pseudotropias aethenoides, using the hypophysation technique. Ramaswamy and Sundarraj in 1956 had done the experiments on catfish, Clirias magur and Hitharpdistis fossilis. But the first successful induced breeding in Indian major carps, Labioruhita and Chihinus mrigala, and minor carps, Chihinus reba and Labiopata, were carried out by Sudhuri and Alikunhi in 1957. Here I should mention that to commemorate their first success of induced breeding in carps, the Government of India is celebrating the National Fish Farmers Day on 10th of July every year. Let us come to the importance of induced breeding. Induced breeding technique is one of the essential techniques for blue revolution, that means revolution of aquaculture. It can fulfill any quantity of demands in any time for the requirement of fish seed for the farmers. It gives pure spawn of certain species of fish. To overcome the mixing of undesirable wild species, it is one of the essential technique. It assures timely available supply of fish seed for the fish culture system. To overcome the uncertain hope of fish breeding, it is very essential technique because fish, the major crabs breed only once in a year during monsoon, and the breeders have to wait till the come of the monsoon in the conventional method. Such waiting is overcome by induced breeding. The technique does not need too much technical assistance. Even a layman can learn the technique very easily. The expenditure for the induced breeding is lower than the natural collections of spawns because in natural collection several manpowers are required. Let us come to the factors influencing spawning in fish. The factors influencing spawning in fish are categorized into three categories, namely physical factors, chemical factors, and biological factors. The biological factors are mostly the hormones. The hormones like follicle-stimulating hormone and luteinizing hormone takes key role for breeding in fish. Out of the various physicochemical parameters, water temperature, monsoon or cloudy weather, turbidity, optimum photoperiod or light, dissolved oxygen, and pH. These factors take key role in breeding of fish. Firstly, water temperature. The water temperature in the range of 26 to 31 degrees centigrade is most favorable for various carp fish. However, the optimum temperature is reported to be 27 degrees centigrade. Monsoon or cloudy weather. Monsoon or cloudy weather is required for more rain for more water current. The water current stimulates the maturation of gonads and more gonadal activity. Turbidity in the range of 100 ppm to 1000 ppm is preferable for breeding in most of the carps. Optimum photoperiod or light is also essential for gonadal activity, gonadal development and maturation. The most important factor for induced breeding in calves is dissolved oxygen. Dissolved oxygen level in the range of 8 to 10 mg per liter is preferable for breeding in carpfish. That is the reason why these carpfish they used to prefer the running environment, because in running environment in nature there, due to the water currents and other factors, there is high dissolved oxygen level as compared to the pond water. Next is pH. pH in the range of 7.5 to 8.5 is ideal for breeding in Indian Major carps. Now come to the types of induced breeding. The techniques of induced breeding can be categorized into following types: band breeding, hypophagation, inducing by natural and synthetic inducing agents. Band breeding is one of the most conventional methods of induced breeding, which is also known as induced natural breeding. But this technique is nowadays is rarely used due to its several disadvantages. We will come to the disadvantages in the subsequent slides. Hypophagation. In hypophagation, pituitary gland extract is used for inducing fish for breeding because Pituitary extract contains follicle-stimulating hormone and luteinizing hormone, which hormones are taking key role for gonadal development and maturity. Then induced breeding by natural and synthetic inducing agents are most widely used for breeding in fish. Firstly, bun breeding. Buns are semi-confined rain-fed seasonal water bodies with riverine physicochemical conditions. Buns are constructed in the vast low-lying area with proper embankments from the three sides, and upper area remain open. It receives large quantities of rainwater after a heavy shower. Buns are categorized into two types, dry bund and wet bund. This is the diagram of a dry bund. You can see these dry bunds are usually shallow water bodies which retain water during monsoon only, and during winter it dry up. Secondly, wet bund. Wet bunds are deeper than the dry bunds and it retains water throughout the year. And each of the bunds they are having an outlet. There is also one third category of bunds which are generally constructed for hydroelectric power generation, that is called modern bunds. Modern bunds is having difference only in the outlet, which is controlled by a automatic opening and shutting down of the gate in the outlet as per the level of water requirement in the bunds. Such type of bunds can be used for breeding purpose also, for breeding of fish. Let us come to the advantages and disadvantages of bund breeding. In the left hand side you can see the advantages of bund breeding. In the right hand side these are the disadvantages. The advantages are major calves generally breed almost at any place of the shallow bunds. That is one of the advantages. Secondly, a few storage tanks, cement cistern or earthen ponds can be provided adjacent to the bunds so that the breeders can keep the brood fishes before releasing to the bunds. But major disadvantages of bud breeding are it is very difficult to coordinate the breeding activity in the bunds. Mostly the collection of spawn it takes lots of labor, and in the vast low-lying area it is very difficult to collect each and every spawn from the bunds. Secondly, there is a chance of getting mix of predatory insect larvae or predatory fish in the bunds since it is near the natural environment. It is out of control of the farmer. Thirdly, the hatching rate of eggs and survival of the hatching are poor. As we know, the fertilization in fish is external fertilization, and in bunds the vast low-lying area is there. In the vast low-lying area it is very difficult to mix or each and every eggs with the milk released by the males, so most of the eggs remain unfertilized also. Lastly, brood fish may carry lots of infections since in bun breeding brood fish are collected from natural environment. In nature there may be different types of infection in their body, and such type of infection may be carried to the pond also, ulcer pond also, where we will release the spawns. Now let us come to the most popular method of induced breeding in fish, that is hypophysation. Fish hypophysation is an artificial method of induced breeding in fish in which the sexually matured brood fishes are induced to breed in confined water like pond by using pituitary gland extract. Since pituitary gland is also known as hypophysis, Therefore, this technique of inducing fish using pituitary gland extract is known as hypophysis. This is the diagram of pituitary gland in fish. This pituitary gland is connected with a bone called sella turcica. It is connected to the hypothalamus. Pituitary gland secretes several hormones, out of which two hormones, namely follicle stimulating hormone (FSH) and luteinizing hormone (LH). These two hormones take key role in gonadal maturity and development in fish. Now let us come to the various steps of hypophysis. Fish hypophysis involves various steps such as selection of breeders, segregation of breeders, stocking and maintenance of breeders, collection of pituitary gland extract, storage of pituitary gland, preparation of pituitary extract and preservation, injection or administration of pituitary extract to the brood fish, and lastly, collection of fertilized eggs and transfer to the hatching hopper. In the subsequent slides, we will be discussing these steps one by one. The first step of induced breeding by hypophysis is selection of brooders for hypophysis. The breeding fishes should be healthy, fully ripe, and of medium sized. The breeders should preferably be in the age group ranging from 2 to 4 years and have a weight averaging 1 to 5 kg. Large size breeders are avoided for difficulty in handling. The most important point for selection of breeders is differentiation of male and female species. To differentiate male and female species, we can see several sexual dimorphisms during breeding season. The male shows roughness on pectoral fin, and when its belly is pressed, milk freely oozes out. In this figure, in the left hand diagram, we can see that males are having narrow abdomen and milk will come out on gently pressing the belly. The right female shows a relatively soft, round and bulging belly, and its vein is swollen, protruding and pinkish in color. Due to the retention of eggs and maturation of the eggs, the female belly become bulging and swollen, and its vein is pinkish in color. Likewise, we can differentiate male and female. We can easily identify. There are some other methods also, like one of the popular method is catheterization. A catheter tube is used usually for collection of female gametes. Eggs are collected with the help of catheter tube to examine gonadal maturity, and such tube also can be used for identification of male and female also. If it is a female, egg will come with the tube, and if it is male, milk will come. Next step of induced breeding is segregation of breeders. Prior to the commencement of the breeding season, males and female fish must be segregated to get optimum activity of the reproduction in the fish. And to do this, before 2 months of the breeding season, males and female fishes are segregated and they are treated to the monosex culture. Now come to the stocking and maintenance of breeder fish. If the breeder fish are collected from the natural environment, they must be collected quite in advance of their breeding season. If it is collected from the fish farm, it is collected 2 months in advance. And they are raised in the breeding or fertilization pool of the fish farm, and they are stocked at a rate of 1000 to 2000 numbers per hectare area, that is in case of carp fish. Suitable measures are taken to keep the breeders healthy and free from infection. Usually, while transferring the breeders from one pond to another, 10% potassium permanganate solution treatment is essential for avoiding the contamination of the microbes, and usually for up to half an hour to 1 hour treatment is done for the fish. The next important step of hypophyton is collection of pituitary gland. The pituitary gland should be preferably collected from fully ripe rabbit fishes. Glands in induced bred fish collected immediately after spawning have also been found to be effective because their hormonal level, follicle stimulating hormone and luteinizing hormone levels are in peak. Most suitable time in India for collection of pituitary gland in major calves is during monsoon, from May to July months. Pituitary gland is usually collected by following two methods. Firstly, through the foramen magnum, and the second method is by dissecting and cutting through the roof of the skull. In this diagram, I am showing the collection of pituitary gland by cutting through the roof of the skull in the labiorhoeta. This diagram in this slide explains the methods of pituitary gland collection for large scale. For large scale collection of pituitary gland, wooden frame is used for holding the fish. Fish is held in the wooden frame and cutting the head with a fine saw or a sharp strong knife. A deal guide is prepared. A cylindrical core of the entire skull in the brain is removed using the cylindrical hole cutter or hole saw attached to the electric drill. A plank with a hole is drilled in. It is kept pressed on the skull to guide the hole saw. The cylindrical core, which is removed from the hole saw with the help of a screwdriver, contains the skull top, the brain tissue connected to the pituitary gland, and the skull. The part of the skull and the brain tissue with the pituitary gland are separated with the help of a scissor, and finally from the brain pituitary gland is separated and picked out with the help of forceps, and the pituitary gland is finally preserved in acetone or ethyl alcohol or subjected to freezing. Let us come to the storage of pituitary gland. Pituitary gland is stored prior to the commencement of the breeding season, and usually three different types of methods. One is cryopreservation method by freezing, secondly in absolute alcohol, or thirdly in acetone. The gland is preserved. Now come to the preparation of pituitary extract. Pituitary extract is prepared by following method. Usually it is very difficult to weight the individual pituitary gland because it is the size of a small pea in a large brute fish of 5 kg. So people used to do collection of pituitary glands together. Several pituitary glands are collected together. Then their dried weight is taken first. After, on the basis of dry weight, so that we can take the extract, preparation of the extract we can standardize. So dry weight is essential. After taking the dry weight using mortars, it is homogenized with the 0.3% sodium chloride solution. In the next step, as per the requirement, the solvent is adjusted. Next step is the centrifugation. After that, the liquid part is used as a pituitary extract for administration. That means pituitary extract can be prepared either in sodium chloride solution or in distilled water. The most important step of hyperphysation is injection of pituitary extract. For each set of breeders for hyperphysation, two males are selected against one female. That means males and females are selected in the ratio of 2:1. Intramuscular injection is most common. However, intraperitoneal injections are also administered in some countries. In India, intramuscular injection is commonly given in the base of the dorsal fin or at the caudal peduncle below the lateral line. An injection is given at 45-degree angle. For injection, clinical needles numbering 24, 22 and 19 are used for the breeders in the size of 1 to 2 kg, 2 to 3 kg and above 4 kg. That means larger the breeder, lower is the needle number. The most important step of induced breeding is the selection of appropriate dose. For females, two doses are usually given. The first is the initial provocative dose, that is 2 to 3 mg per kg of dried pituitary per kg body weight. That means if a breeder is of 1 kg, that means for 2 mg injection we have to give 2 mg. For 2 kg breeder 4 mg injection, then for 3 kg breeder 6 mg injection. And the second injection is given at an interval of 6 hours, that is higher dose, that is 5 to 8 mg per kg of the body weight of the female. And males are given a single dose of 2 to 3 mg per kg body weight of the fish, and that injection is given at the time of the second injection to the female. And sometimes what happens, despite of the two doses of pituitary extract to the female fails to breed. In such cases, a higher dose can be administered, that is around 8 to 10 mg per kg of the body weight. Or due to some problem called co-clogging of the eggs, we can use striping method. The striping method is conventionally used for collection of eggs and sperms with the pressing of the abdomens, and in striping the male and female gametes are mixed with the help of quill feeders, and that can help to fertilize. That is done actually if the hormonal injection fails to release the eggs or sperms. Next step is breeding in hapa and spawning. After the injection, the brooders are released immediately inside the breeding hapa. A breeding hapa is generally made of fine cloth in the size of 3.5 meter in length, 1.5 meter in breadth, and 1 meter in height for large breeders, and for small breeders smaller than 3 kg, it is in the size of 2.5 meter in length, 1.2 meter in breadth, and 1 meter in height. All the sides of the breeding hapa are stretched and closed except a portion on the top for introducing breeders inside. Generally, one set of bitters is released inside the breeding hapa. After the release of the fish, the opening of the hapa is securely closed so that brooders may not jump out and escape. Instead of hapas, cement cisterns or plastic pools as big as hapas can also be used for breeding. Spawning normally occurs within 3 to 6 hours after a second injection, that is effective dose. Soon after fertilization, the eggs are swelled up considerably owing the absorption of water. Fertilized eggs of major calves appears like shining glass beads of crystal clear transparency, while the unfertilized ones look opaque and whitish. The size of the egg from the same species of different brooders varies considerably depending upon the size of the brooders. Fully swollen egg of the Indian major calves measuring 2.5 mm in diameter, the largest being that of Katla and smallest of Rohu. The calf eggs are non-floating and non-adhesive type. The yolk possesses no oil globule. The Indian major calves have a profuse egg-laying capacity. Their fecundity on an average is 3.1 lakh in Rohu, 1 to 3 lakh in Katla and 1.5 lakh in Mrigal. The developing eggs are retained in the breeding hapa undisturbed for a period of 4 to 5 hours after spawning to allow the eggs to get properly water hardened. After this, the eggs are collected from the hapa using a mug and transferred into a bucket with a small amount of water. The brooders are then taken out and weighed to find out the differences before and after spawning. This gives an idea of the quantity of the eggs laid. The total volume and number of eggs can be easily calculated from known volume and the number of eggs of their sample mug, which is known as volumetric method. Percentage of fertilized eggs is also assessed according to the conducting random sampling method before and after spawning. Random sampling can give us an idea of the quantity of the eggs laid. Let us come to the advantages and disadvantages of hyperphagisation. Fish hyperphagisation is having several advantages, such as high quality of pure fish seed can be obtained at required number, and huge number of fish seed from 5 lakh to 1 million can be obtained depending upon the size of the fish or species of the fish. Maximum growth rate can be produced by selective breeding technique while doing the hyperphagisation. Several breeders can be bred at a single location, and most important advantage of induced breeding is that same female fish can be used more than 2 times in a year, which is not possible by natural breeding. In natural breeding, fish breed only once in a year, but in hyperphagisation technique, 2 to 3 times in a year. These are the disadvantages of hyperphagisation technique. There is a chance of fish getting injury. Since female broodfish get more than one dose, then usually 2 or 3 doses during the time of induced breeding, there is a chance of getting the injury of the female fish during holding of the fish. And there are limitations of effectiveness in cross species. That means the pituitary gland prepared for a species may not be effective for other unrelated species. And most important disadvantage is that as per the requirement, the required quantity of pituitary gland is not available and it is not readily available in the market. Now let us come to the most advanced method of induced breeding in fish, that is use of natural and synthetic inducing agents. Several natural and synthetic inducing agents are successful for breeding in various groups of fish. These hormones or these agents are very popular among the fish farmers. Some of the natural inducing agents like human chorionic gonadotropin. Human chorionic gonadotropin is the hormone secreted by placenta during pregnancy. This hormone can be extracted from the urine of the pregnant woman. Cynahorin. This is nothing but chorionic gonadotropin and mammalian pituitary extract mixture. This cynahorin is found to be effective in various carp fishes as well as catfishes. For the induced breeding in carps, the most successful and popular agents are synthetic agents in the modern context. Several popular synthetic agents are overprim, pymozide and LH-RH analog, DOCA, that is 11-deoxycorticosterone acetate, 17-hydroxycorticosterone. Out of these, this overprim is most widely used and most popular synthetic hormone, which is successfully used for breeding of various group of fish. Overprim is a dopamine antagonist, which is readily available in the powder form and that can be used by the fish farmers. The advantage is that it can be used only as a single dose. Most of the synthetic hormones, they require only single dose for breeding of male and female, unlike pituitary extract, which is required 2 or more doses for inducing the female. The data presented in this table shows the differences between induced breeding by hyperphysation and induced breeding by synthetic hormone. You can see doses of pituitary extract versus overprim. In this table, three major carps, namely Lebiocatla, Lebioruhita, and Chinas migala are shown for the use of pituitary extract and overprim. For breeding of female of the Lebiocatla, Lebioruhita, and Chinas migala, a preliminary dose of 2 to 3 mg per kg body weight is used and higher dose or second dose is 6 to 8 mg per kg of the body weight. That means 2 doses are required for inducing female for breeding. But in case of overprim, you can see that one dose of 0.4 ml per kg to 0.5 ml per kg is effective for Lebiocatla female. Lebioruhita 0.3 ml per kg to 0.4 ml per kg. In case of Chinas migala 0.25 milliliter to 0.3 milliliter per kilogram of the body weight. In case of male, single dose is required for both the hormones, pituitary extract or overprim. But in case of the females, 2 doses are required for pituitary gland hyperphysation technique, which is somewhat problematic for females because females have to be catched for 2 or more times for induced breeding by hyperphysation, which is overcome by the use of synthetic hormone. That means synthetic hormones are having advantages, more advantages than the pretty realistic. To sum up the presentation, we can say that induced breeding is the most significant advancement in the field of aquaculture, in the field of blue revolution. Out of the different induced breeding techniques, namely band breeding, hyperposition, and use of natural and synthetic inducing agents, hyperposition method is popular among the fish farmers. Hyperposition involves administration of two separate doses for female brood fish. However, most preferred inducing agents for breeding in fish is the synthetic hormones or synthetic agents, which are required to administer as a single dose and most preferred by modern fish farmers because they are readily available. Only single dose is required. These are some of the references which I have consulted during preparation of this presentation. For knowing the details of induced breeding techniques and various advantages and disadvantages, you can go through these references. If you have any queries regarding this presentation on induced breeding techniques, you can contact me with the email chittaranjan_21@yahoo.co.in. I hope that through this presentation, at least you have some preliminary idea on various induced breeding techniques and their advantages and disadvantages. So thank you for listening.